Our last session of the cell and development last talk of the cell and development session is by Dr. Sheena Fraser. Sheena holds a PhD in biotechnology from Rhodes University and has been working with countless biotech and cultured meat companies. She's currently the VP of product development at Roslin Technologies, and today you will be talking about scaling up iPSCs. So I'm very much looking forward. And
all right, absolutely. Thank you for the introduction. Yes, so Sheena Fraser. I head the product development team at Roslin Technologies. I'm supported by a team of 20-odd stem cell scientists. We have a very small new team of bioprocess scientists that we're building in-house at the moment, and really trying to translate and transition our cell biology expertise into an industrial mindset. How do these cells grow in the kinds of reactors that our clients and customers are using out there? So we at Roslin Technologies, stem cell biologists, we sell and produce iPSCs and embryonic stem cell lines to the cultivated meat industry. They are selected across a range of livestock species. Most of our experiences is with our porcine and our ovine iPSCs. We do have embryonic stem cells in the pipeline as well, with bovine coming to our sort of fore in the next couple of months and being made available, with seafood cell line development happening in the background. So today, our iPSCs, the porcine iPSCs, is really where I'm going to be focusing most of our attention from a scale-up perspective. Here we use Sendai technology to derive those cells, so it's non-integrative RNA vector system where the Yamanaka factors are used to reprogram those cells into a pluripotent state. And we really go through a process of screening these cell lines quite heavily, on the upfront side, making sure that they meet a series of quality standards. We look at their potential to differentiate because they are pluripotent stem cells, so we really need to make sure that they meet those criteria and maintain the stability in industry, that they are karyotypically stable as they are expanded, and that they retain the potential to differentiate into multiple lineage and cell types going forward. We have got a poster outside on our adipogenesis differentiation, poster 11. You can have a look at the advances that we've made in adipogenic differentiation on our pluripotent stem cell lines. There is also, we use a lot of RNA sequence analysis and looking at the differences between embryonic stem cells and induced pluripotent stem cells, and how we can harness that knowledge to improve our understanding of derivation going forward, and how we can translate that into media improvement, process improvement, and development. My business unit at Roslin Technologies, product development, is very much focused on media optimization. All of our cell lines grow in serum-free media, but the knockout serum replacements that are commercially available are quite expensive. We need to replace those formulations with food-grade regulatory compliant solutions, and ultimately make sure that whatever media we're developing and the conditions that we're using for our cells can be scaled, can be used to increase high cell densities and really improve the efficiencies of the process in an industrial perspective. What I'm going to talk about today is really just taking a step back at the fundamentals of process development. We look at how our cells behave in benchtop systems, because that's easier for us. We're not a large-scale manufacturer. We rely on collaborating with our industry partners for that. We look at how our cells respond in a 2D environment, mainly 6-well plates. We also take that through from a 2D environment, your adherent growth, and look at how they respond as in suspension culture, and how that changes as you change the reactor geometry and the scale of the solution, the technology going forward. So from that perspective, we've got a good starting point. The induced pluripotent stem cells that we're working with, the porcine iPSCs, are really stable. This data set is for one of our newer lines that are coming into our pipeline. Other data takes our population doubling levels up to over 400 times, but this particular cell line shows stable growth and stable pluripotency, the main pluripotency markers over time in the culture environment. And that helps me from a process development perspective, because it really means I'm starting with a good cell line. I'm starting with a really good material, and that is something as well that you can carry through into industry, is your product is only as good as the ingredient you start with. And how you translate that and then build on that is how you end up with a really efficient commercial process going forward. Now, if we look at how that changes when you move from 2D into suspension culture, it's actually really interesting what we found and what we saw. We track our cell line pipeline development through the seed trains. We monitor growth and stability through the life cycle of, as you're scaling up sufficient cells to seed a suspension culture, and then how those cells themselves respond in that liquid suspended environment. And at a small scale, even at a small scale, should I say, we see not stable pluripotency, but we see an increase, an enhanced pluripotency coming through with these iPSCs. And we're not really sure why that is happening. It's most likely due to the microenvironment that the cells are exposed to. And there's a lot that we need to unravel when it comes to spent media analysis, the oxygen gradients, the mixing efficiencies, the shear, everything that happens in that environment is going to impact cells, and our understanding of really what we can do to manipulate those process conditions to produce a more efficient and scalable, robust and scalable protocol going up and forward. So these cells grow as aggregates. We don't need microcarriers. So they aggregate and they form these spherical spheres of cells that are kind of difficult to control the size and the shape. But if you manipulate those process conditions, you can see how you can adjust the mechanical forces of the reactor, the aeration, the oxygenation, to manipulate those conditions to change that profile a little bit. I'm going to dig a little deeper into the OCT4 going forward here. I'm not going to highlight the rest, just for simplicity, so that we can track that marker as an indication of pluripotency going forward. Is one of the most, I suppose, closely monitored pluripotency markers when you're looking at stem cells. So it's a good one for us to track as we go forward. Before I move on there, I just want to highlight what we're seeing here with these iPSCs is doubling times as low as 13 to 16 hours. So it really is remarkable how we can transition from 2D environment into suspension with very little changes to the fundamental properties that these cells have. They really are efficient at expanding and growing, and we want to maintain and stabilize that as we actually increase scaling capacity. So we then move on and we scale up from 6-well to Erlenmeyers, 125 ml Erlenmeyers, into spinner flasks, into reactors. So we follow this transition of what one would be a seed train in most production environments from that point of view. And we look at fed batch, and moving on to continuous fermentation is the end point, is how do we get to a point where we can maximize cell densities and then maintain those continuously going forward. And this is all where we are moving towards from a product development and support point of view. When we scale up, we see the same pattern. We see pluripotency increased over time. So here, and the viability of the cells is maintained with the high agitation and shear rate and a lower oxygen condition and oxygen transfer in the spinner flask because it's not gassed. We actually see a higher pluripotency coming through there, and translates to slower growth. So we see a growth slowing down of 21 hours in these environments, predominantly because of the oxygen and the nutrient mixing and balance from that system. And then these spinner flasks are not pH controlled as well, so you've got that build up of metabolites and waste that impact the cells as well. And moving forward, it's finding out what the ammonia toxicity levels are, lactate toxicity levels are, and how we can rebalance the media to improve growth and performance. When we then take that next step further, scaling up to 3 liters in a less efficient environment, this particular reactor was run in collaboration with Flex by the iPSC at Heriot-Watt University. They retrofitted a microbial reactor and refitted it to operate mammalian cell culture, just as an indication of what can be done in industry with the tools and the systems already exist there. So suboptimal conditions, but we saw really good growth. The aggregates got really large. The shear stress and the forces in that particular reactor allowed the cells to grow quite big, and that had a significant impact on viability going forward. But once again, pluripotency was increased. So what is really happening here? Are we seeing necrosis in the center of these aggregates? Probably. Are we seeing nutrient gradients across the dimensions of those aggregates that give you different profiles from a pluripotency perspective and give you that optimal, ideal condition that if we figure out what that gradient is, we can hone in and target the specific parameters that will give us the best performing proliferation going forward. Scaling down again to 500 mils, Applicon system again, this system designed for cell culture. We see a far higher shear in the reactor dimensions at a small scale than we did at the larger scale 3 liter version. Understanding the differences in those design parameters and how those impact the process conditions is something that we need to model now. But what we see in the smaller scale system is we see that we can actually use shear to break up those aggregates into more of a single cell like suspension. We can change the media conditions to promote a more dispersed growth and improve the nutrient delivery to those cells. And in this case, we actually see a much higher increase in pluripotency with these particular cells. So there's a lot of work that we still need to do here. But it really is exciting to see that these iPSCs can grow robustly under a number of different conditions. And whatever you do to them, they seem to adjust and recover, and they're quite resilient. And I think that's where I'm quite excited to continue working with these going forward. In summary, today, over 100 doublings. I think we're working a stable cell line. It's a good starting point. We've seen a number of presentations today which show that after 48 hours you're getting senescence. You can't develop a process like that. You can't optimize conditions when you're doing things like that. We've seen immortalization, genetic engineering of cell lines to create a stable culture going forward. And the recombinant events, the reprogramming events of induced pluripotent stem cells seem to induce a similar kind of response as well, that reverting back into that stem cell-like process. Doubling times of 13 to 16 hours, really that is going to be significant improvement in process economics down the line. If we can maintain that and pluripotency and the differentiation potential of these cell lines as they scale up, then we have got a competitive process that can be commercialized at scale. The trick and the challenge then is how do we differentiate them in that environment? Pluripotent stem cells are more difficult to differentiate than primary cell lines. They're further back in the lineage. But we are making significant advances in that. You'll see from the posters that I referred to earlier how we've actually managed to direct the research to improve the timelines around that. And media improvement. You can't sustain a high cell density culture in an optimal state without following the tenets of spent media analysis and process design going forward. What's happening here? I'm still unraveling it, trying to figure it out. The modeling those different reactors, I think, is going to play a key role, and really just challenging and testing the boundaries of the process parameters going forward, particularly the normoxic, hypoxic environment we know is important for stem cell culture. Now, how do we translate that into process? And in summary, we don't do this on our own. we hope to learn from the people that we work with, our collaborators, our partners, our academic research collaborators, as well as industry. And the more we understand ourselves, the better we can actually make sure that cultivated meat is enabled. And yeah, I think taking it from there. Okay, thanks.
Thank you so much. What a great talk. Thanks. Are there any questions for Dr. Fraser?
Thank you for the team.
Over there?
Thank you. Okay, that was an easy catch. Thank you so much for a really interesting talk, Dr. Fraser. Do you think that with the Applicon half liter, did you check the LDH? Was it possible that the viability was higher because some of the cells were sheared instead of, being poor viability? Sorry for the feedback.
It's entirely possible that some of those cells are shared. Breaking them up, you're going to see that. We didn't see significant foaming, so it wasn't that much shear happening in the reactor. But at the same time, we can't rule it out. And from that point of view, yeah, work in progress.
There's another question up here.
Perfect. Thank you for a very nice talk as well. I noticed you mentioned you had one passage point in your 3D data. Can you share a bit more of, are you able to maintain them and passage the cells? I'm thinking in particular for seed trains that you want for upscale production.
So from a seed train perspective, because we work at small scale, we model that by going from 3 milliliters to 40 millimeters in 100 ml flasks to the 400, 500 ml in a reactor up to a 3 liter. So we monitor it from beginning to end through that point of view. The largest scale that I showed here didn't go through the full cycle. It was a tea flask that went into a 500 ml that went into a 3 liter. So that information is captured. The data I show is we start the passage that I show starts at when it first goes into suspension.
Okay.
Is what this data shows.
Okay, thank you.
Okay, maybe one last question. There in the back. Let's stay local.
Thanks for the presentation. I think in the previous slide you showed like the results between Applicon 3 liters and Applicon 0.5. And I could see that the aggregates looked completely different. Do you have an explanation why the difference between those two?
Yeah, so the distinction between those two is that the 3 liter one was a microbial system where the dimensions of the impeller width and depth and the vessel volume were slightly different. So the shear properties were not as high. That reactor was also run at between 100 and 120 RPM in that environment to keep the cells suspended. Mm-hmm. They were maintained in suspension at those agitation rates. When we applied those same conditions to the smaller scale, the 100 to 120 RPMs, the aggregates settled out. So we had to increase the agitation, and through that process, we saw dispersion. So there's a lot of factors that are changing between those systems that we need to figure out what exactly we can control going forward.
And what do you think then is the effect on those cells that are supposed to grow on aggregates to be released to the media and be just single cells?
Well, from monitoring pluripotency, it actually looks like the dissociated cells retain a higher average pluripotency for the cell population than the aggregates. I think that you're going to see different nutritional gradients across the dimensions of those aggregates when they grow as spheroids, and that is probably going to introduce culture heterogeneity, which will also impact the performance and the efficiency of differentiation downstream. So in many ways, although an aggregate culture stabilizes mammalian cells, it may not be the most efficient process at scale going forward. I think a dissociated culture might be more efficient and easier to control, particularly the differentiation step.
Okay, thank you very much.
Thank you very much.